Everyone to Wednesday night at the lab. My name is Dylan Brewer and I'm a student worker here at the Biotech Center. On behalf of the Biotechnology Center, UW Madison Division of Extension, Wisconsin Public Television, Wisconsin Alumni Association, and UW Madison Science Alliance, welcome to Wednesday night at the lab. We do this every Wednesday, 50 times a year. Tonight it is my pleasure of introducing Su-Tran Zhang, and tonight he will be sharing about human stem cells and how far we've come in 25 years. Before we get started, we're going to ask him a couple questions. Where were you born?
Ask that question. I was born in China.
All right. Where in China?
On the southeast coast of China.
All right. And where did you attend high school?
In my hometown, that very little town, village town, very small town.
All right.
Yeah.
And where did you go for undergrad?
That's in Wenzhou Medical University. So it's a town a couple of 100 kilometers away from my home.
All right.
Yeah.
And what did you study while there?
I studied medicine.
All right.
That's my under.
And where did you go for any advanced degrees?
Yeah, I did my master degree then in Shanghai Medical University. So that's another city. Probably many of you know that city.
Mm-hmm. All right, everyone, please join me in welcoming Su-Tran Zhang tonight.
Thank you, everyone, for coming in the winter time. Because this week is somewhat special, and so I'm going to talk about the topic that happens sometime 25 years ago. So relating to the topic, I'm going to mention stem cell research at UW Madison. And I'm also going to tell you where we are. So why I pick up this topic, as I mentioned, this week, 25 years ago, you might notice, some of you, that in our university, we developed the first human embryonic stem cells on this campus. And that piece actually is written by Terry David. And it goes like, the dream of one day being able to grow in the laboratory an unlimited amount of human tissues for transplantation is one step closer to reality. So why is that? Because at UW Madison, the person who did that was Jamie Thomson. Many of you probably have heard of him. He developed the human embryonic stem cells in his laboratory here at the Primary Center, University of Wisconsin-Madison, for the first time in the history. And I want to also mention that he actually did many other type of stem cell. One is monkey embryonic stem cell a few years back. And that actually set up the foundation for him to develop human embryonic stem cell technology. But as I'm going to tell you later on, that several years later, he developed another type of stem cell called induced pluripotent stem cell. I'm going to touch a little bit later. Why the development of human embryonic stem cell caused such a kind of a splash? This is because, the stem cell comes from this little place in the early embryo. It's called the inner cell mass. It's a cluster of cells consisting of a few hundred cells. And these cells actually, in the end, produce any kinds of cells in our body, from skin, bone, muscle, to brain cells, all the type of cells. And so these are called stem cells. And what the Jamie did was taking this group of cells and then grow them in a petri dish. and they grow like that as a cluster of cells. And this cell can be grown pretty much continuously until today, actually. Today, the cells we are using were actually initiated by Jamie 25 years ago, more than 25 years ago. And so you can imagine that how many cells we can actually produce over that many years. And plus, because of their potential, the capacity to give rise to any kind of cell, you can imagine that what the utility would be. So at least at that time, it was imagined in this way. I just want to show you one example. For example, the cells, the stem cell grow in a petri dish, and you will see they are beating. You almost can immediately tell what they are, right? They are heart-beating cells. And if you count, it actually beats around 60 times a minute. Why is that? It means that the cells grown in the petri dish, they actually remember who they are, what they should be doing. And if we take, let's say, the same type of stem cell from a mouse, the embryonic stem cell, and they differentiate to heart cell, they will beat how many times? Almost 10 times more, like 500 times a minute or something, because they are coming from mice. And from human, they beat around 60 times a minute. So that means they remember what they should be. And so if we take stem cell, guide them to become other type of cell, we probably would also be able to do that, right? So, and another part is that the cell, I mentioned that we can continuously grow them. You know, the cells can divide once a day, for example. 2 days will be 2 cells. 3 days will be 4 cells, you know, and they keep going. And generate a huge number of cells for any kind of application. You know, has its potential and capacity, but there is also coming with an issue, because the cells were initially coming from an embryo, even though it's in vitro fertilized embryo, the couple no longer need them and they are going to discard it. Still, you know, there are ethical issues. One is the people who against it would be, okay, killing this helpless human embryo for scientific research represent an assault to, you know, respect for human life. But on the other hand, there is also a counter-argument that it's immoral not to use to pursue research on otherwise discarded embryo that has the potential to find treatment and cures for world's most devastating diseases. And so there are always ethical arguments surrounding the embryonic stem cell research. And I took this Time magazine page. Just you probably noticed many of the faces, right? And because of this research having these ethical issues, and the time the research on human embryonic stem cell were not funded by any federal government. And so in order for the cell to be utilized for research purpose and for developing therapeutics, we need the support from all aspects. And among those faces, you probably notice one person we are very familiar with, right? Yeah, so Jamie Thomson. another Thomson. He was our former governor and the former secretary of Human Health and Human Services in the Bush government. And he was very instrumental in persuading Bush government to allow stem cell research go on. And he came to visit us many times to understand what this stem cell research entails, how the potential it would be, and so on and so forth. So in the end, I think he persuaded the Bush government to allow the research on human embryonic stem cell for at least the stem cell lines that were already generated by that time. But still, at that time, the funding, federal funding for research in that area was not allowed. And so that's why the university and the Alumni Research Foundation, many alumni like many of you are here, and help us to promote the research. The first thing was done by the Wisconsin Alumni Research Foundation to build the non-for-profit research institute called the Y-Cell Institute. It started with several of us. So for that, I want to remember one person, it's Carl Brunson, and he was the director of WARF. He really championed for the stem cell research here at UW Madison. And that institute was designed to promote the stem cell research, in particular at that time, to allow the building that was not contaminated by federal fundings, and that was pretty tough at that time. And so that really made stem cell research here at UW Madison move forward. But it goes way beyond that. We trained a lot of people how to grow those stem cells, research on those stem cells. Not only UW, not only in the United States, but also people from around the world. And furthermore, with that institute, we actually shipped cells to all over the world. Okay, and just to give you some idea, over the past 25 years, we shipped cells now over 10,000 shipments to over 3,000 investigators in 900 institutions, more than 48 countries in 6 continents. So you can see how a small medicine can contribute to the world in that particular area. But still, that is not sufficient with just that institute. So we actually, the university, through communication with many entities through public communication, and went to Capitol Hill, talking to people, and we even invited many of the celebrities. I mentioned here, it's Michael J. Fox. You probably know him. He has Parkinson's. He promotes stem cell research. The upper one actually is our former governor, Jim Doyle. Many of you know them. And so many people actually help to promote the research. So in the end, I think besides at the beginning 2001, when the stem cell, the existing stem cell lines were allowed to do research, later on, Obama administration further rules remove the remaining restrictions, so allow the stem cell research to move forward. So I want to tell you this brief history because, this kind of initial discovery unnecessarily went through this bumping road getting to where we are today. But another important part in science is that we have those embryonic stem cell research going on. But that research actually led to another further discovery, that is development of another stem cell type called induced pluripotent stem cells. And that was done by two people. One is Shinya Yamanaka in Japan, as well as our old friend Jamie Thomson here at UW. And Shinya actually received a Nobel Prize for that. Yeah. So you see how the embryonic stem cell research here leads to the further development of another type of stem cell. And this why this induced pluripotent stem cell, short for iPS cell, why they are so important. Because these cells can be generated from any of us here. You just need to donate a drop of blood. We can generate such kind of stem cell. So it means every of us can have a stem cell bank in our cabinet, right? And yet those stem cell iPS cells are very similar to the ES cell, the embryonic stem cell. They can proliferate continuously. They can generate all kinds of cells in our body. And of course, has less ethical issue because it's generated from our own body. And it also means that this cell can be used for personalized regenerative therapy, because it's coming from our own body, right? So with this kind of development, our school also established another center. It's called the Stem Cell and Regenerative Medicine Center to further promote the research on both embryonic stem cell and induced pluripotent stem cells. And what this center is aiming at to advance science in stem cell research and also foster regenerative therapy and maintain the forefront status of stem cell research here at UW Madison. Okay, so I have introduced to you what stem cells are and the brief history of stem cell, particularly from embryonic stem cell to induced pluripotent stem cell. So next I'm going to tell you, what do these stem cells do? So I will just give you a couple of examples from what we are doing in our laboratory. So I'm working mostly related to brain functions. So I will tell you a few things relating to stem cell in terms of neurological or psychiatric disorders. So one thing, in order for the stem cell to be utilized, the first thing we need to do is to guide this stem cell to specialized cells. So in this case, I'm showing you is the nerve cells. So we can actually develop technology, guide the stem cell to nerve cells, either in a two-dimensional, or we can actually make them almost like a brain-like structure. We call it brain organoid. And recently, we even further developed a technology to 3D print them. So because we have stem cell, we can guide them to become many kinds of specialized cells, and we can actually take those specialized and print them. And so this is a piece of brain tissue. And they are alive, by the way. And they talk to each other. And so, and with those specialized cells, now you can do a number of interesting things. You can look at how our human being is developed in early time, because we could not study in the past. We can only use animals, right? Now we can actually look at directly. The second is if we take the cell from patients, or we genetically modify them, and we can actually look at how they change in the disease process. And if we have that, we can also use this as a platform to test the drug on those patient cells directly, not necessarily going through animals. And finally, we can actually have this cell as a source to replace the cells that are injured in our body or lost in our body. And so as a cell therapy strategy. Okay, so I'll give you one example. For example, we can guide the stem cell to specialized cell, motor neuron, it's called motor nerve cell, meaning the nerve cell controlling our movement. And that research was initiated by an ALS patient when he called me from Texas over the phone saying that, Dr. Zhang, you need to work on this. And I said, I never worked on that, but it doesn't matter, you have to work on this. And I actually started working on that project. It turns out that we were still the first in the world to develop, to guide the stem cell to become motor neurons. And this is just one example. And over the years, actually, in my lab, we have guided the stem cell to retinal cell, brain cell, many different kinds of brain cells. I listed over a dozen of them. These are all specialized, and those cells are either injured or diseased in many of the disorders like retinal disorder, Parkinson's, Huntington, Alzheimer's, ALS, and so on and so forth. And this is just from my lab. But there are many other labs at UW Madison. And so in our campus, actually, there are many other types of cells generated. For example, I mentioned the neural cell, but there are blood cells, vasculature cells, cardiac myocytes, and so on and so forth. So what I'm trying to say is we actually have developed the technology to guide those stem cells to many different kinds of cells in our body, from skin, muscle, bone to brain cells. Now, how, because we have so many type of cells, so now we can actually make it very standardized platform, like using 3D printing. I just want to show you one example. We actually recently developed a technology to print a piece of tissue layer by layer. This is brain tissue, and they can actually talk to each other. And if I give you a 3D kind of look, you can see this nerve cell with nerves connecting to each other. And I use a different color to show different type of nerve cell. They actually handshake each other, talking to each other, essentially. And so these are all alive. And so we can really begin to understand in our how these nerve cells each other. And I mentioned that if we take cells, the stem cell generated from patients, for example, if some patient or any of the people give a drop of blood, and we can generate these iPS cells, and then we can guide them to, let's say, nerve cells. And say, give you an example. If I take ALS patients, ALS, probably many of you have heard of it. It's also called Lou Gehrig disease. Those patients, the nerve cell, the motor neurons died, and so they get paralyzed, and in the end, they could not breathe, and they died from that. And see, when we took those patients here, and then look at the nerve cells we generated, and you can see this is from patients. And those nerve cells, you have swelling of this called the neurofilament, it's the cytoskeleton of the nerve. And because they swirl around, instead of the normal one, they line up in parallel. So that really jammed the nerve, so they could not transduce the signal along the nerve. And that really shows you how powerful, when you take a patient and you look at them. And you can also use this technology to look at some of the unknown diseases. We recently discovered a family of patients in Middle East. And this family, the kids in that family show this premature aging. When they are kids, when they are teenagers, they actually show this aging sign, gray hairing, skin, white color skin, all these kind of things. And then they also have very severe neurological and the cognitive deficit. These kids, when they get to teenager, they could no longer move and become paralyzed. What happens to this? And this disease, up until today, when I'm talking to you, it's not diagnosed. And when we took a skin punch and generate this iPS cell, and then guide them to brain organoid, this is a piece of brain tissue-like structure. And you see the top one is the normal one. Under the lower row is from patients. And you probably can tell, I specifically using color to label different cell types. You can tell the difference. The cells at the bottom, the green cell, many fewer, not lined up evenly, right? These are dividing cells. They're supposed to divide to produce enough cell to form the brain. But those kids do not. Yet, when we look at the upper part of the brain organoid, you see the normal one is that thin, yet the patient are much thicker. So that means they prematurely generate a nerve cell. So they divide, but they prematurely become nerve cells. So that's why those patients have a smaller brain and have a defective brain connection. So that's why those patients have neurological problem and cognitive deficits. And so you can see that we can use this technology to even help us diagnose those diseases and potentially come up with a strategy for therapeutic development. And with this kind of stem cell platform, particularly those from patients, we can actually modify to a platform for drug testing. Just give you one example. So I mentioned that we can take a drop of blood and from a patient and then generate iPS and then guide them to stem cell and then screening. And for example, we develop a platform for SMA, spinal muscular atrophy. It's also fatal diseases. When babies have such kind of mutation, and their muscle, the nerve cell controlling the muscle, do not function appropriately, so they cannot move or they cannot even suck milk and so on and so forth. And so it's usually a fatal disease. And we actually developed this platform and screening drugs in collaboration with NIH. And you see this is the reference drug, and actually identify several of them. And later on, in clinical trial. So it's quite powerful in identifying drug because you can test directly on patients' target cells. And so for that reason, I created a small startup several years ago in response to the call from the university. It's called for Discovery to Product program, D2P program. And so we developed this human nerve cells for drug company or biotechnology company. Look at the disease process, look at the drug development. So we can produce now many different type of cells I listed here. And not just me, there are many others. And so I took a statistic from Wolf. Over the past 2 decades, Wolf licensed, patented many of the discoveries here relating to stem cell technology and licensed them, and accumulated the income is $50 million. But that's just for the licensing, and it mostly targeting these well-known farmers and biotech companies. But it also created the startups within medicine area, and I listed a few of them. These are the list of the companies built here within medicine. So far I've told you that this stem cell can be used as a technology platform for looking at the disease process, for screening drugs, right? You still have not heard from me that in the first page that the stem cell will be used for therapeutics, right? So I'm going to tell you a little bit in the last part on that part. So cell therapy. So how can the stem cell be used for treating many debilitating disorders? When, of course, you can take the cell, generate the specialized, let's say, nerve cells, and replace the cells that are lost in the disease. But you can also use these cells as a vehicle to carry molecules that are missing in our body. So that's the second way. The third way is to remodel the environment of the liver or spleen or kidney or brain to promote the regeneration. Finally, in the brain in particular, you still need to, not only the cells you need to replace, but also you need to reconnect or rewire the network. So it's much more complicated. And I mentioned that with the iPSC technology, use the pluripotent stem cell technology, we can cell from our own body so that we can actually use these cells for personalized therapy, because these are coming from our own body. So if you use it, they will not be rejected. Not like many of the transplantation many of us have heard, let's say kidney transplantation, you have to use immune suppression throughout, right? But here, if we use our own cell, so you don't need the immune suppression. So I will give you one example. Let's say many of you have heard Parkinson's disease, right? It's a very common neurodegenerative disease. And this disease actually is caused by degeneration of the dopamine nerve cells in the midbrain. And this nerve cell degenerated, and so our movement is not controlled, so have trembling, instability, and so on and so forth. Now, we have treatment options. We can use medication like L-DOPA, or using surgical approach like deep brain stimulation. And they are actually quite effective in improving the symptom, to minimize the symptom. However, all these treatments actually do not stop or even slow the progression of the disease. So the disease still progresses as is. So in the end, those medication or surgical approach, they lose their efficacy. So that's why we need a new approach to deal with this kind of disease. And so cell therapy is one potential possibility. And this area actually quite interesting because in the past, actually over the past few decades, people have tried to use fetal tissues to treat Parkinson's disease. Some are effective, some are not. The reason is because you cannot standardize the treatment. Plus, there is ethical issues. And so if we have the stem cell, now we can actually guide them to become, let's say, dopamine nerve cells. And we can standardize them. Then it becomes a drug, right? A medication. And so that is what we have been doing over the past many years. And we actually have developed a way to guide stem cells to these dopamine nerve cells. And I use many panels because these dopamine nerve cells are very specialized dopamine nerve cells. They have to have certain kind of features, including the expression of those genes, and not other genes. And when these cells are transplanted back to the mouse brain experiment, we can only use mouse. And so let's say we transplant the cell into the mouse brain, also midbrain area, and then you ask, what this nerve cell do. These are the human stem cell derived dopamine nerve cell. And you will see here in the brain section, and that's where we transplanted, and the dark area are the nerve, where the nerve goes. And you see the nerve goes to all the way here. This is a magnified view. It's straight in the area that controls our movement. And that is what exactly this nerve cell do in our brain. It's just mind-boggling because those nerve cells are generated in a petri dish. And you put back into the mouse brain, not even human brain, they know where to go. They actually find their way, find their home, and reconnect the neural network. And so that's why this animal actually recovered. Their motor deficit is mitigated. But in order to demonstrate that this motor recovery actually is coming from the transplanted cell, we actually engineer the stem cell before transplantation. We build a switch into it. So how do we do that? And so we can turn on or turn off the function of the transplanted cell. Let's see if we turn off the function of the transplanted cell, so the therapeutic effect will be lost. And so it will be indicated by the mouse turning to the left side. And so we remote control the mouse, turn off the function of the transplanted cell. So when you let the mouse move freely, and you can see it always turns to the left. So that means it loses the effect of the therapeutic function. But if you want the cell to function more, and you can beat them, beat them to work harder, and then what's your expectation? It will go to the right side. This is just the readout, and so we turn the positive switch, they turn to the right side. And so that demonstrates that this cell indeed do what they are supposed to do. And because of that, we are moving to the preclinical, to a larger animal, because you can cure hundreds of thousands of mice, but it doesn't necessarily mean it will be effective in human, right? You have heard so much in news that the mice are cured, but when we move on to human, that's another story. And so we decided to move on to the larger animals using the non-human primate. And so how did we do that? So we took a skin punch from the Parkinson's monkeys and generate the stem cells from them and guide them to dopamine nerve cells and then transplant them back into the same Parkinson's monkey. And this was done in collaboration with the Primate Center and the imaging done at the Weizmann Center. Now, and we follow those animals exactly like we follow the patients. Do all the tests on patients, on those monkeys in the exact same way as for patients. And you see, this is just the PET imaging to look at whether the transplanted cell release dopamine or not. So the top part panel is the animals that before giving transplantation, you can see the right side is empty, right? The left side is red. Now, the lower panel is the transplanted, after transplantation. You can see that the right side, there is a signal on the right side of the brain. So that's the transplanted cell release dopamine. And when we look at the behavior symptom, and those animals actually recover, and they move freely. And so this set of animals tells us that, hey, this approach can be safe and can be also effective, actually. And more importantly, because those cells are coming from the same monkey, so that means it's autologous. So we actually didn't use any immune suppression. After surgery, they are free, And also because we did a number of animals, so we can actually come up with many of the parameters so that we can use those parameters for application in patients. And so that's why we are now moving toward the patient application. Now, the final part, how do we move from the lab to the clinics? I mentioned briefly that nowadays we can use different approach, either from your own cell or from somebody else. So I will just use your own cells. Let's say you take a drop blood, generate stem cell, And then we can generate many different kinds of cells, right? For, let's say, blood cell, you can use it for blood disease like leukemia and so on and so forth. You can generate the insulin cell, for example, for diabetes. Maybe retinal cell for eye disease, or the nerve cell I mentioned for many of the neurological disorders. Just give a couple of examples. One is the blood cells. I took Dr. Igor Slakvin's lab slide, and they actually developed a technology to guide the human stem cell to many, many different kinds of blood cells, from neutrophil, macrophage, T cell, NK cells, these lymphocytes. And this cell can be used to treat many of the leukemia blood disorders. But he's also developing technology to help avoid immune suppression. Because when we use cell transplantation from one person to another, this is called allogeneic transplantation, which would need immune suppression. And sometimes it can create the reaction, because the immune reaction to the transplanted cells. And it can create the so-called graft-induced reaction. And he's now developing the mesenchymal cells from those stem cells, and to regulate this response so that can minimize the risk for this kind of cell transplantation therapy. And they have already, gone through the phase 1 clinical trial. At least it shows that it's safe through his company, Sinatra. And another part is retinal cell, for example. It's from my colleague next door to my office, Dave Gam. He's in ophthalmology. So they actually generated the retinal cells from the stem cells. And just give you one example, this panel, the middle panel, is the retinal cells generated from stem cells in a petri dish. And the right side is the real retinal cell in our retina. Look at them, how similar they are. Of course, this is just by morphology, but more importantly is functionally, they are actually very similar. And so that's why Dr. Gam also established a company translating this technology to clinical application. They are also in preparation through FDA approval for clinical trial. I mentioned the Parkinson's experiment I did over the past 2 decades, and I felt that we have overwhelming data to support the move for clinical application. So that's why I also set up another startup, Brain Cell Therapeutics, to initiate the clinical trial. And this is the new company, and the first part is to work on stem cell. And in this case, we will use autologous transplantation. Basically, very much like what I did on the monkey studies, taking patients' cells, guide them to dopamine nerve cells, and transplant them back. And so that's what we expect to get in approval from FDA next year, and hopefully start the clinical trial a year later. But we are also working on a number of other diseases which we do not have actually any treatment options. One is spinal cord injury. Because when when the person is injured the spinal cord, they are paralyzed, we do not have any options actually. And so over the years, we actually developed technology to turn the stem cell to specialize the spinal cord cells. And they can regulate the pain, movement, and so on and so forth. And we have already tested in rat models, and it worked well. And we are also testing in the monkey model, at least from MRI, showing that they can integrate into the spinal cord. So we are now in the preclinical stage. Hopefully we will get to the patient sometime. Another also very devastating disease is stroke, and it's also very widespread disease. And when there is a stroke, the brain is damaged, and they cannot regenerate. And so we have developed a way to guide the cell to the brain cells, but still there are many hurdles in this part, because if you transplant cell into the lesion, the stroke area, the cell actually could not survive at all. And so we recently developed an engineered cocktail. Hey, with that cocktail, the cell survived. You see the blue area, this is the lesion area in the stroke area. And with the transplantation, you can fill the stroke area completely, reconstitute the brain. And so we are now also moving to the larger animals before we move on to patients. I kind of give you only a few examples over the course, but I just want to summarize that the first stem cell, human stem cell, were generated here in Madison 25 years ago. And that leads to the newer version of stem cell. It's called iPSC several years later. And it looks like it takes very long time for us to get to where we want to go. Indeed, it takes very long time to develop technology, particularly in terms of how to guide the stem cell to produce specialized blood cell, pancreatic cell, nerve cell, and so on and so forth. But now, if I look at what's happening, particularly after 2020, in this area, you see the first patient trial in Parkinson's patient was in 2020, and then in the spinal cord injury patients, and then the clinical trial for Parkinson's in many other areas in China, EU, US, Canada, Japan, and so on and so forth. And also recently there is also a report showing that the cell therapy is effective for epilepsy. And these are just the neurological disorders. And there are many others you already have heard. Diabetes, for example, macular degeneration, and so on and so forth. And those things happen actually mostly within this past few years, right? A couple of years. So you can see where the stem cell research is heading. It really gets to the critical stage toward the clinical application. So that's my prediction. But before I end, I just want to come back to tell us how proud we are here. What UW has done has impacted the field, particularly in terms of stem cell research and regenerative medicine. And I mentioned that it's the place, where human stem cell originate. But I also mentioned that in the historical perspective that it went through multiple steps, how it impacts the policy funding for stem cell research. And I mentioned that we actually, through this process, we actually trained a lot of people worldwide in distributing cells across the globe and stimulate and nurture the stem cell investigators, including also industry. And in the end, it will benefit people who suffer from many of the debilitating disorders. So I will stop here by acknowledging a lot of people because the work I've done in the lab, I mentioned, done by my students, postdocs, undergraduate researchers, and so on and so forth, and of course also from many other labs. And the funding support from federal and non-federal private industries. I mentioned many things not happening in my lab, and those are coming from many of the colleagues who provided all this information. And I also mentioned the 2 companies I set up because they are actually very important to take the technology developed in my lab to application in potentially in the future in clinics. Okay, I will stop here taking any potential questions. Yeah.
How do you guide a stem cell to more function?
The question is, how do I guide the stem cell to specialize the cell, like a nerve cell, right? So that's a very good question. That's something we have been doing for the past 2 decades. So one is to follow the principle, we call it the developmental biology principle, we have learned from animal studies. So basically, how the cells, one type of cell you are interested, how this type of cell develop during embryonic development. And by using these principles or the molecular pathways we learned from these studies, and then apply to the stem cell technology, stem cell biology. And we, of course, sometimes it works well because they are very similar between animal and human. Mostly are similar during early development, but sometimes are not. So that will require us to figure out or how the in human this cell develop, and then we will be able to come up with a strategy to guide the stem cell to the specialized cells. Does it make sense to you?
Oh, wait, back here with the microphone. We're going to pass the microphone around so that we hear the questions in the video.
Okay.
So thanks. So the question I have is, so you talked about differentiating the stem cells, but you also mentioned that stem cells, when they're injected into the body, they know where to go. Can you elaborate on that, or is not, or am I oversimplifying that?
You do a little bit. So we did not inject stem cell directly to the body or to the brain. We usually guide the stem cell to the target cell or the specialized cell. In our case, specialized nerve cell. For example, for Parkinson's disease, we actually guide the stem cell to become dopamine producing cells and then transplant to the brain. So we do not transplant the stem cell directly into the brain because that could be problematic, because the stem cell, we already discussed that they can give rise to any kinds of cells. So I don't want the stem cell to be in the brain and they produce bone, hair, or other type of cells. I only want nerve cells. So that's why we usually guide the stem cell first to the cell type you want, and then do transplantation.
And you put it in, but you have to put it in a specific part of the body even after, once it's differentiated.
Right. Because because for the brain disorder I mentioned to you, for Parkinson's disease, you need to put the cell where they need that type of cell, that type of molecule produced by those cells, right? And so you do not put it anywhere else. So it has to be specialized, right? Okay.
Yeah. So can you give an example of what you actually do to guide this cell? I mean, in the embryonic development, there are chemical gradients and things that
Okay.
happen, but what's an example of what you actually do to this dish of
Very good point.
native
Just like
stem
you are
cells?
making cocktail, right? You put gin or Coke or something. That's what we do, actually. So you are right. For example, we are trying to guide, let's say, stem cell to motor neurons, which are damaged in ALS patient, for example. And so what we do is we take stem cells out of the content that will maintain them in the stem cell state. So we change the condition, the medium, the cocktail. So let's say they normally like to drink red wine, for example. I no longer give them red wine. I put them into just plain water. And so they cannot maintain their state, no longer be able to maintain them as stem cells. So they are going to become something else. So now I need to add something else to induce them to the nerve cells, for example. So, for example, one molecule that is important is called retinoic acid, basically vitamin A, right? And then they actually turn into the spinal cord cells. And then there are a few other steps you want to guide them, not only the spinal cord cell, but also spinal motor neurons. And so you need to add a few other molecules. One is called a sonic hedgehog. That's a good name. But what it does is to guide those cells to become motor nerve cells. So it's a step by step. You have to coach them. Very much like what we, take our baby, gradually crawl, sit, walk. Each step you have to give a very specific kind of guidance. And in this case, we give molecules, small molecule or chemicals to guide them. And each step, we use different combination of these chemicals. And those are learned from what you just asked from developmental biology. These chemicals normally regulate the development. And so that's what we apply to the stem cell. One at a time, usually it's a combination.
So it's a series of changing the solution that the cells are growing in.
Right.
You have to add different things to it at different times.
Right, exactly. You got it, you can do it now.
How effective can stem cell treatments be overall in treating genetic diseases where the cells that you're creating might also still have those genetic defects that lead to the disease in the first place?
Terrific point. So what if I have a mutation and you have the cell and you put it back, you still have the same thing, right? Now, there are a couple of ways of thinking that issue. One is that you can correct the mutation. It's very easy. Nowadays, with this technology, right? We actually do it routinely. Pretty much everybody in my lab can do, right? You take patient cells, you know the mutation, we can just correct it. you have heard the name called CRISPR, right? It's a technology. We can just correct it, or we can actually make it a disease model. That's what we do in the lab to create a model system to understand the process, right? So the approach is to correct the mutation, and then you do all the steps I mentioned. You correct the mutation and guide the stem cell to the specialized cells, and then do transplantation, right? So another way is that in some cases, in actually many cases, it may still work because many of the diseases develop over decades. So let's say Parkinson's disease. Even some of the patients have mutations, it still takes many decades to develop. So in that case, even if you do not make a correction, the cell can still be functioning for many decades. It may still work. But the ideal way is to correct the mutation first, right? And you do not worry about this mutant cell will create further problems. Okay?
So over the past 25 years, we've made like a lot of progress, as you just talked about. Do you have any sort of guess or like inclination of what you think is going to happen in the next couple of years, or what you think the next like big breakthrough or something along those lines is going to be?
Yeah, I kind of hinted in the graph showing you that in over the past 25 years, the first 20 or something years are mostly focusing on this research development kind of area. But in the recent 2-3 years, you can see many of those things actually begin to move into clinic, at least in clinical trial. And in response to your question, my expectation is that in the next few years, you will see explosive kind of trial taking place in many, many diseases. And of course, for drug development, it usually takes quite a while because of the regulatory requirement. You have to go through phase 1, phase 2, phase 3 kind of trial. So that is the timeline you can expect. But I expect that some of this stem cell drug will, in the end, be utilized for treating many of those diseases.
You've talked so far about remedial treatment of disease. Is there a place for stem cell treatment as a preventive and to make us stronger, have stronger immune systems and things like that? And if there are obstacles, are they more than just technical?
I think it is also a great area of interest nowadays because indeed we can learn from this fundamental stem cell biology. And then this kind of information we learn from those biology can actually help us how to keep the cells young, not to get too old or too aged, or at least stay healthy. And so this kind of research actually is undertaking now, actually. And hopefully, such kind of information will help us prevent some of the debilitating disease, particularly degenerative disease, because degenerative disease, whether it's arthritis or Parkinson's, it's usually related to age and wearing kind of over time, right? And what if we learn from this stem cell biology, and we can apply this kind of information to maintain the cells in the healthy state and slowing them from going aging kind of process. And so that can be actually preventive. And some people are thinking of anti-aging type of strategy, right?
What do you view as the biggest hurdle facing the stem cell industry right now in getting the cells into humans for actual treatments?
Very good question. I face a lot of problems. One is the science part. I think we actually have made great progress, and I think that part actually we can always overcome as a scientist. Now it comes down to the drug development. The drug development, you face a number of hurdles. For each drug to be developed in Western country, in the US, for example, costs $1 billion. Where do you find that money? I couldn't. I have a hard time. So that's one. Two, regulatory requirement. Because I think somebody already asked, stem cell, they can generate any type of cells. So you want to be absolutely sure that when you have your drug product, you put into patient, you create no harm. So meaning that you do not have other type of cell that could potentially create harm, right? And so that's why FDA has a very strict requirement. So you have to do very repeated, very solid demonstration that you created no harms. Yeah. And so that's the second regulatory hurdle. And then I think these are probably the two biggest hurdles I'm now facing, actually. There are many other smaller ones. Those can be overcome.
So do you see this treatment becoming accessible to all people, or do you see it becoming a standard form of treatment? Like when you think of cancer, you think of things like chemotherapy. In the future, could you see stem cell treatments as accessible for all people, or more of an expensive option or something?
That is a very, very good question. I think it's kind of related to the earlier question I brought up. Because of the requirement by the regulatory body, and because of the expense that is needed to develop a drug. And also, this drug is very different from the traditional drug, the chemical drug. This is a living drug, and it's very tough to develop. And so the cost usually would be pretty high. And that gets to your question, whether that kind of treatment can be accessed by everybody, right? So that question is difficult to answer at this moment because it's not there yet, because it's related to the regulation, related to the insurance, and all sorts of things. It needs to come together, all these elements, in order for everybody to be accessible to this type of treatment. From our perspective, of course, we would love to have this to be used for everybody, right? Because that's our goal. But the reality sometimes is not as simple as that. Even not for stem cell therapy, even for many other of the drugs developed nowadays. But in the end, I think it will require, scientists, regulators, government to come together to solve these kind of issues. And I think you raised a very great question. It needs to be solved by the time. But I think people are working on it, probably. Yeah.
Okay, down in the front.
How is research being conducted in other countries which may not have the same regulatory barriers that the United States has and maybe have different funding models?
Yeah, I think you actually raised a good question, and it actually happened. You know, in different countries, they have a different regulations. Particularly at the beginning, when embryonic stem cell technology started, some countries do not have such kind of restriction, and so let's say in Asian country, they just move very quickly. And when iPSCs started, because it was initiated in Japan, so Japan actually took it almost like a national pride. So they push very hard, and so that's why some of the clinical trial I mentioned actually started in Japan, and the government give a lot of funding to promote that part. And we here are mostly coming from private funding. So it's a different model system. You know. On the other hand, we are still an innovative country, and so we are still doing well. So it's, yeah, indeed there are different model systems going on, regulating or influencing the stem cell research and regenerative treatment development. Other questions?
Yeah, any more questions? All right, thank you so much for an amazing talk.
Thank you.